Now, what I'm going to do is review very briefly some of the ongoing projects and some of the future projects that are being done in our Comparative Genomes and Biodiversity group, which is run by Bruno Sobral, who's here and has been carried out in his lab currently at the California Institute of Biological Research. We're going to talk a little bit about his work on sugarcane, which is an enormously important crop that almost no one in this room has probably ever seen. It's the largest biomass producer of any crop on Earth, and it's probably the fourth or fifth largest economic engine in the world at converting sunlight into money. Now, what we have here is a situation, as you'll see, where sugarcane is an enormously difficult crop. Basically, like grad students, it almost never has sex. Wow, things were different when I was a student. We have an enormously complex octoploid, as you'll see, and basically there has never been genetic improvement of any substance in there except by truly empirical methods. What we want to do is see, can we use new genetical methods based on arbitrarily primed PCR to allow us to not only map the heredity and pedigree of existing sugarcanes, but make predictions how we can do sensible and intelligent breeding to diversify the sugarcane populations. Next, I'm going to very briefly describe how cassava, which is another crop most of you have never seen, which feeds more than 500 or 600 million people in the world, can be optimized by looking at historical pedigrees from centers of biological diversity. We'll talk a little bit about bananas. Again, not much sex in any of these. Tropical crops tend to have problems with that. And then finally, we'll talk about the genetic resource indexing technologies group that we're trying to put together to overcome the logistical and technological barriers to doing far more sophisticated analysis in situ in the tropical countries. And in doing so, I'm going to bring to light what I think is a very, very compelling concept of hologenomes, of the fact that the individual is a dead concept. You drop a mouse in a blender and there's more bacterial genomes in there than there are mouse genomes. And I think that they're as or more important than the mouse nuclear genome in the mouse's performance in nature. And that's going to affect agriculture enormously. Not that a mouse is an agricultural beast. These modest, easy to work with little potted plants in a glass house in Texas are actually, again, a simulacrum of the real world. This is in Papua New Guinea. This is the real stuff, the wild accessions of sugarcane. And if we're to look at the history of cultivated species, we have to go out and look where it's not cultivated and look at its history, what little is preserved anyway. This was in a collecting expedition in 1928, and you can imagine how daunting it is to be faced with an enormous population of undomesticated canes and saying, what is it? How do you take samples? What is out there? The differences between dealing with species that clearly have sexual diploidy and those species that have very little sex and highly heterozygous polyploids is almost impossible to describe to those who have not experienced it. But back then in this famous expedition, collecting expedition, what they found is all canes were intercropped with taro, with cassava, with a variety, including bananas, of other crops in extraordinarily complex systems that have been used for over 1,000 years and had been continuing on what are otherwise considered fragile tropical soils to produce. That's sustainable. But there are certain other difficulties faced by people working out in the field in tropical climates. National Geographic drew attention to it here. Seeking disease-resistant sugarcane, scientists find Neolithic man in unmapped nooks of sorcery and cannibalism. This is, of course, by the principal pathologist in charge of cannibalism. The next slide shows such a circumstance. No, I'm sorry. Sorcery maybe, but not cannibalism. This is Bruno, Rhonda Honeycutt, and that's Mike. Is that Mike? Yeah, Mike McClellan. And then some of the F1 from Bruno. Basically, what their lab did was set out to use an arbitrarily primed PCR approach to try to look at developing a genetic map for a very, very difficult crop like sugarcane, where even the basic ploidy was not established initially when this work was undertaken. There was no cytogenetics of quality. Sugarcane chromosomes cytogenetically look like little dots. So they had to take an approach using arbitrarily primed PCR, which was co-developed by Mike McClellan and John Welsh at the CIBR at the same time as the DuPont group developed it. And the basic approach was to use primers of arbitrary length to prime PCR, obviously, in different parental types. And there was a great deal of optimization. This just shows one example slide of Bruno's work in the amount of work that was required to optimize for arbitrarily primed PCR. Now, I should emphasize one of the big challenges in environmental and agricultural samples is that the DNA is of very limited quality and very difficult to get. Making sugarcane DNA is extremely difficult, but it's much easier than most tropical plants. Nonetheless, getting reliable DNA preparation proved to be an extraordinarily boring but really important bottleneck. Nobody likes to say they spent 3 months just to make their template prep, but in fact that's what often happens in agricultural systems. Optimizing for not only primer concentration, type of primer, type of enzyme fragment had to be done, but once it was possible in a very small amount of time to do what tropical agricultural biologists thought was largely impossible, which is to develop a fairly dense map of the sugarcane. This has been published. It is now more than double this density, but basically something that no geneticist could ever conceive of because there are no visual markers in sugarcane at all. And this has been used to start making substantial pedigree analysis for existing canes and starting to look at breeding strategies that can be incorporated. A byproduct of this work, I should point out, of Bruno's was the discovery that virtually all sugarcane in the world is a monoculture for chloroplast, which is terrifying to the breeding community. About 20 years ago, the maize community, the corn breeding community, was faced with a disaster of enormous proportions when mitochondrial monoculture of American corn turned out to be susceptible to a particular fungal disease, and it wiped out, I don't remember the number, but a very, very large proportion of American corn production. And the idea then of genetic vulnerability became widely spread in agricultural circles. So this is also true for sugarcane, which much of the tropical world depends on for economy.